Hello, everyone. Welcome to today's live broadcast titled Automated Optimization of IgG Production in CHO Cells, presented by Dr. Mike Kowalski, Staff Application Scientist, Beckman Coulter Life Sciences, and Dr. David Apiyo, Marketing Applications Manager, ForteBio, a division of Pall Life Sciences. We are excited to bring you this educational webinar presented by LabRoots and sponsored by Biologics by Danaher. Biologics by Danaher is a combined team effort from select Danaher companies, Molecular Devices, Beckman Coulter Life Sciences, and Pall ForteBio. Combining years of experience and scientific knowledge, they provide comprehensive biologics solutions with the simplicity of a single point of contact. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we begin the presentation, we have a few important announcements to make. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. Now, I'd like to introduce Dr. Mike Kowalski. Dr. Kowalski is a staff application scientist in the Drug Discovery and Basic Research Group of Beckman Coulter Life Sciences. He received his Ph.D. in microbiology and molecular genetics from Harvard University. Our second speaker today will be Dr. David Apiyo. Dr. Apiyo is a Marketing Applications Manager with Pall ForteBio in charge of manufacturing, bioprocess, and QC applications. David received his Ph.D. in chemistry at Tulane University and held 2 postdoctoral stints at Rice University and at the Pacific Northwest National Lab. For complete bios of both of our speakers, please visit the webinar registration page and click View Bio. Mike, I will now turn it over to you.
Thank you, Julie, and thank you everyone for attending today. As Julie mentioned, we'll be talking about some of our collaborative work where we automated the optimization of IgG production, and we'll show how this fits into a larger CHO cell line development workflow. A brief agenda for the talk today. We'll begin by introducing the process of cell line development and some of the steps involved. We'll focus on one specific step, which is media optimization for improved IgG production. And we'll describe how we automated a design of experiment approach to try and find the optimal conditions for media growth. We'll also touch on how we use multiple different analyses and how this is a portion of how this work became so collaborative. We will focus on analysis using the Pall ForteBio octet for IgG production. And then we'll show how we use an imaging system and a plate reader from Molecular Devices to measure cell growth. We'll also talk about some of the caveats that we found during this analysis, and then we'll bring it all back to show how this fits into the cell line development workflow and how these products can be integrated into a complete cell line development system. So what is cell line development? Well, obviously the goal is to basically produce high quantities of a protein of interest from cultured cells, which is much easier than, sounds much easier than it actually is, and we'll walk through some of the main steps of the workflow. The first step is to basically get your cells to start producing your protein of interest, and this is typically done through transfection, although there are obviously other ways to introduce your plasmid of interest. Following the transfection, there's usually a selection and or enrichment step to basically ensure that all your cells have taken in the plasmid and are now starting to express your protein. Following this, this population is then distributed to isolate single cells. This can either be done using a cell sorter or some sort of limiting dilution approach, because you want your population to start from a monoclonal population and ensure that all your cells have the same makeup. Following this step, you need to basically do a screening assay to assess the protein production that each well or each colony is currently producing, with the hopes of finding not only the highest producer, but also the ones that are producing the molecule that has the correct characteristics. Once this has been completed, you would take your top hits forward and then see if you can further optimize the process to increase protein production even further. Once these optimal conditions are established, that's when you would move to a scale-up and move towards a bioreactor type size of protein production. Now, there are a number of challenges associated with this workflow. The first would be sample throughput. Not only does sample throughput change dramatically throughout the process, but at its highest points, such as during the clone selection portion, you can have significantly high throughput, and it's essential to have sample preparation and analysis systems that can meet this throughput. You also have a concern for sample and data integrity. This workflow can take place over weeks or even months, and you need to be able to ensure that your samples are known where they are in the system, and any data that has been generated is associated with those samples, and we have some hardware and software solutions to enable this. And finally, as I mentioned, these campaigns can take weeks or months, and the sample throughput, because it can change so radically, really lends itself to trying to stagger multiple campaigns on top of one another so that you're really maximizing the equipment that you have set aside for utilization. And we, again, have some software solutions for this challenge. For now, we're really just going to focus on one aspect to kind of do a deep dive, and we're going to focus on the process optimization step and show how these different components can be used for this step. As I mentioned before, this is essentially pretty far down the stream of cell line development, but what we're hoping to accomplish is once we have a stably expressing cell line, to see if we can optimize the media conditions to really improve our IgG production even further. And the approach we took to demonstrate this is a design of experiment, or a DOE approach. Rather than just looking at changing a single variable at a time, the advantage of DOE is that you can combine different reagents, different concentrations of reagents, different times of addition or incubation, or really change any sort of variable that you can think of. And you would do this in combination with changing other variables. The reason for this is that you can readily identify what factors have a significant impact on your system. You can also identify what factors do not have a significant impact, and you can stop spending time or money or effort trying to optimize these factors further. Ideally, you'll also be able to detect some interactions between factors and possible synergies that can eventually lead you to identifying the optimal conditions to push your system to its maximal level. The challenge with this approach is that you quickly end up with large numbers of conditions because every time you add a new factor, the number of factors that you're testing increases factorially. So things can really spiral out of control very quickly. Our approach for this was to use a stably expressing CHO cell line, CHO DP12, which is expressing human IgG against interleukin 8. The basal media that we were seeking to optimize was the OptiCHO media, which already has been optimized for CHO cell growth, but we were looking to see if we could push things even further and increase the protein production from our stable cell line. For this, we chose to test a variety of different additives that are commonly used in cell culture, particularly in protein-producing cell culture, such as polyamine, Spite, which is a mixture of selenium, pyruvate, insulin, transferrin, and ethanolamine. Glutamax, which is a more temperature-stable form of glutamine, a frequently added amino acid. Non-essential amino acid solution. 2 different buffers as a way of trying to normalize the pH if we did have increased cell growth. Those were bioreactor pH adjustment solution. We'll refer to it as bioreactor buffer from now on, as well as the more commonly used HEPIs. And one more additive, efficient feed A, which is made by the same manufacturer as OptiCHO, and again is suggested to help increase CHO cell growth. The conditions we were testing were simply yes or no adding these different additives. So even with just these essentially 2 concentrations plus or minus the addition, already the combinations reached 96 different mixtures simply from this relatively small number of additives. If we actually increased that to add a second concentration so that we had no addition, high and low, the factorial nature of this experiment would result in 1,215 different combinations. So you can see how quickly this can spiral into pretty difficult numbers to work with. In addition, we wanted to have some replicate wells to ensure that we had reliable measurements. And you can see because of this 96 different mixtures plus replicates, this would definitely be a workflow that would benefit from automation. That brings us to the Biomech automated workstations for Beckman Coulter Life Sciences. Shown to the right is one of our Biomech I-Series instruments, the I7. This is basically just a liquid handler that has a variety of different functions. One pipetting head is the span 8, shown on the lower left-hand side, which has 8 individual pipetters that can be spread apart so you can access multiple tubes, and can also aspirate and dispense different volumes at the same time. Shown in the lower center image is the multi-channel head. This can be either a 96 or a 384 well head, and those can be changed between methods. And it's also shown featuring our selective tip feature, where you can load a single column or a single row of tips, a single tip, or even clusters of tips, really any combination you can think of, and we'll show how this was really essential for this DOE approach. And shown on the lower right-hand side is the deck of the instrument. The deck is designed to be flexible, so you can have positions that can hold tips or plates, but you can also have active positions that can heat and cool, that can shake, or you can also integrate different devices. An example of which is shown in the upper right-hand corner. The Biomech itself is enclosed in a HEPA-filtered enclosure, and this was used to ensure the sterility of the samples while we were manipulating them. You can also integrate such things as incubators, shown on the far right-hand side of the integrated system. This enables the cell to be brought on and sent back to the incubator without manual intervention. And on the left-hand side, you can integrate an analyzer, or really on either side, as well as on the deck itself. And shown on the left is just one of the analyzers we use for molecular devices. So this gives you an idea of the system that was used. We'll now walk through the workflow itself and how we approach the design of experiment. So shown here is just a 96 deep well plate where we started using this to combine the different factors that we were testing. We use the span 8 system to add either media, indicated by the minus signs, or spite or polyamine, shown in blue and red respectively. And as we established from the DOE approach, we had combinations of all of these reagents resulting in 4 different conditions to start. We next used the span 8 to repeat the process using media, glutamax, or non-essential amino acids, shown in green and orange respectively, and plated those into 16 different combination wells, and then used the selective tip feature of the multi-channel head to load 4 tips and stamp our original 4 wells into those 16 wells in quadruplicate. So we now have 16 different conditions of the combined factors. This step was repeated, again using the span 8 to dispense either media, HEPIs, or bioreactor buffer, shown in yellow and pink outlines respectively. And we loaded 16 tips on the multi-channel head to, again, stamp those original 16 conditions into our new 48 wells. We then brought on a second 96 well deep well plate and dispensed either media or efficient feed A into 48 wells each, loaded 48 tips onto the multi-channel head, and stamped that for a total of 96 different conditions. We then loaded 96 tips onto the multi-channel head and dispensed them into quadruplicate wells of a 384 well plate. Again, this adds challenge to trying to do this manually because 384 well plates are a challenge to work with. But you can get an idea here of what the plate layout looks like. And the 4 highlighted wells really just indicate the 4 quadruplicate wells that happen to feature HEPIs, glutamax, spite, polyamine, non-essential amino acids, and efficient feed A. So again, in a single plate, we're able to test 96 different conditions in quadruplicate wells. And my best estimate to try and do this manually was that it would have taken about 184 manual transfers using both single and multi-channel pipettes to do this process that the Biomech was able to achieve in 20 minutes. After all of these reagents had been dispensed, we added cells to each of the wells and incubated them for a variety of days, after which we would need the opportunity to try and assay the production of IgG to see which of our factors was having the most impact. And for that, we turned to the octet from Pall ForteBio, and I'll turn it over to David to tell you more about that.
Thank you, Mike. So just a brief introduction of the octet systems, that were used to monitor IgG expression over time. There are 4 main octet systems that differ mainly through different throughputs. So we do have the low throughput octet K2, which is a two-channel instrument essentially capable of measuring two molecules simultaneously. The next level of the octet systems would be the third generation octet red 96E, which is capable of measuring simultaneously 8 samples at a time. In addition to the octet 8-channel instrument, we do have the octet red 384, which is a 16-channel instrument and therefore capable of measuring 16 samples at a time. And then finally, we do have the octet HTX, which is Pall ForteBio's flagship octet instrument, capable of measuring as many as 96 samples at the same time. The last two instruments, the octet 384 and the octet HTX, are configured to be able to be integrated to a liquid measuring device such as the BioMech. So the main difference in terms of the sample input for the instruments would also include the design of the sample plate and the biosensor plates. So both the octet red 384 and the octet HTX have two different sample compartments where two sample plates, either a 96-well plate or a 384-well plate, can be inserted. In addition, all the instruments do have a biosensor racking plate where biosensor racks containing as many as 96 sensors, as you can see in the schematic in the diagram, can be inserted. A sensor, biosensor, which essentially are single devices used for measuring each sample at a time, are essentially fiber optic materials that are made also of a glass tip, at the end of which there is a bioactivated molecule. So Pall ForteBio carries a variety of these biosensors with different biomolecules at the tip, covalently linked, including protein A and protein G, which are essentially used for measuring IgG titer, depending on the different isotypes of the IgG. In addition to the protein A and protein G, there are also other customizable biosensor tips, such as streptavidin, such as amine reactive tips, etc., depending on what kind of analysis one wants to run. So in addition to the biosensors, what's required to be able to run an assay on the octet instruments would simply be a sample tray, a 96-well or a 384-well sample tray. So the technology behind the octet systems are real-time label-free biosensor analytical systems that utilize biolayer interferometry. Essentially, when we talk about biolayer interferometry, what we mean is that the system measures the light interference as you build up biological molecule at the tip of the biosensor. The biosensor itself, of which a schematic is shown in the diagram on the left-hand side, has two surfaces, one an internal reference surface, and the second a chemistry sample surface at the tip of the biosensor. The two surfaces reflect light independent of each other and deflect them back to the detector. So essentially what the system does is that as you build up biological molecule through the chemistry surface on the tip of the biosensor, when you're binding a molecule, you essentially have two different light reflections going back to the detector that are captured remotely in the instrument and then are used to be able to then determine what the interference pattern would look like as a result of the building biological molecule. So the system then captures the intensity of the light reflected as a function of both wavelength and the distance between the two surfaces, essentially the reference surface and the sample surface. So you can imagine as your concentration changes or as your molecular weight size changes, you essentially expect the intensity to change and also potentially the shape of the wavelength of the sample waveguide to change. The system therefore shows the difference between the lambda max of the reference waveguide and the sample waveguide in real time. So what you see on the diagram at the bottom is what the user, the analyst, sees as the analysis is going on in real time. And so for any given binding event where one expects an association between two molecules, you should see a shift, a red shift in the wavelength that ends up giving you a positive response signal. If, on the other hand, one wanted to look at the quality of the reagent being analyzed, one may potentially then move the sensors back into buffer and be able to look at the dissociation rate of the same molecule. So in addition to looking at binding events, one can also look at dissociation events. In the case of IgG quantification, the analysis basically stops only at the binding event phase and does not necessarily proceed to the dissociation phase. So with regards to this particular experiment that we did, essentially a quantitation assay is one where you are only looking at the binding event as depicted in the figure in the middle of the graph at the top, where you can see a positive response on the y-axis, the shifting binding wavelength as a function of time. There are three different types of quantitation experiments that can be done on the octet. You can either have a one-step quantitation event known as a direct quantitation, where the biosensors are dipped into the sample in only one step and measured, or you could have a situation whereby the sample being analyzed is fairly low in concentration, typically in the sub-microgram per ml concentrations, where a sandwich assay is required. In a sandwich assay, what would typically happen is that the biosensors are dipped into the analyte and then dipped into a detection reagent, similar to what one would do in an ELISA sandwich. But one could also extend the analysis to include the presence of substrates, especially when you want to measure very low sub-microgram per ml concentrations, such as picogram per ml concentrations. At the end of the analysis, what's then done is that the system then uses the binding events and analyzes the initial on rate of that binding event and plots that as a function of the sample concentration to generate the binding rates of the different concentrations in a standard curve, a reference curve. The reference curve is therefore then used to then extrapolate what your unknown sample concentrations would look like. But in addition to just IgG quantitation or any given molecule, biomolecular concentration determination, the octet can also be used for target ID determination. The octet can be used for lead screening assays, lead characterization, preclinical development assays, and even QC and manufacturing assays. So for this study in question, as Mike mentioned earlier, the cells were incubated for a period of between 2 to 4 days. In general, the IgG quantitation or the expression level determinations can be done throughout the time course of the assay. So whether the cell culturing is, takes, as low as 2 days or as many as 1 week, one could potentially be able to aliquot the samples at any given time and be able to fairly quickly measure the IgG concentrations in a direct assay if the expected expression levels are high, or in a sandwich assay if the expression levels are extremely low. In the case of IgG and in the case of this study here, protein A biosensors that bind to the Fc region of the IgG were used, and the octet HTX, which is the 96 biosensor, 96 channel instrument, the highest throughput octet instruments available, was used to allow for a fairly quick analysis. The samples were initially, first of all, transferred using the automated biomech workstation at 50 microliters into black tilted bottom plates. Biosensors are also initially prepared by being preconditioned in regeneration reagents. So again, because we're dealing with IgG here, where we know that when an IgG binds to protein A, we could potentially use low pH buffers to be able to dissociate the IgG's binding. We can use low pH buffers of 10 millimolar glycine at pH 1.7 to be able to do the regeneration and neutralize the buffer in either neutral pH or in the actual base system that's used for the IgG expression. The entire time spent for preparing the samples, essentially transferring them into the plates, was as low as only 7 minutes. So 7 minutes to prepare the entire 384 samples into a sample plate. The time for doing the actual reads was only 20 minutes for the entire 384 samples. So in this case, because we were expecting a relatively high concentration, so above microgram per ml, a simple direct read was done here, and that took only 20 minutes for the entire 384 samples. And so in a typical IgG quantitation, the determination of the reference standard curve is the first step, and that typically involves using a known standard sample that is similar to the analyte being quantitated. The standard sample is diluted in a dose response titration, so in this case it was from 50 micrograms per ml to 0.78 micrograms per ml. What's important here is that the standard curve or the reference curve needs to cover the span of the expected expression levels. So our estimation was that we needed expression levels of above 0.78 micrograms per ml, but potentially below 100 micrograms per ml, hence the range of 0.78 micrograms to 50 micrograms per ml. The next assay would then be to dip the sensors into the unknown samples. So the diagram you see at the bottom left would be what the 96 unknown samples look like at the different DOE conditions. So again, the idea is to ensure that the curves for the unknown samples fall in between your lowest curve for the standard samples and the highest curve for the standard samples. And then ultimately, the initial slope for the standard curve samples, are analyzed to extrapolate or to determine the binding rates, which are plotted on the y-axis as a function of the known concentrations on the x-axis. So the dot in blue would be your standard curve, binding rates as a function of concentration, and the ones in red are the 96 samples, which are the unknown samples. So as you can see, they fall very well in the middle of the curve, and therefore the standard curve can be used to extrapolate the actual concentrations of the unknown. So we then used, after determining the IgG concentrations, we used a statistical analysis. Essentially, we used a statistical software. The particular one in this case was the Design Expert 8.0 Start, but one could potentially use any statistical software such as JUMP to be able to look at the interactions between the different conditions. So for example, the idea here was to identify what the positive factors in the cell culture supplements would be, and also to identify what supplements would potentially be negative factors which would not necessarily promote IgG expression, and then finally to identify what the optimal conditions for IgG maximization would be. So the first graph you see, and these are just examples of some of the interactions, based on the analysis. The first graph you see is an analysis of the interaction between glutamax and spite. So what we can see is that when you add spite to your cell culture media, the base of cell culture media in this case was OptiCHO media. The red lines are the glutamax conditions. Over time, we do see an increase in the IgG expression over time with glutamax. But when we add spite to the same media that has glutamax, and that's depicted in the green lines, we now see a reduction in IgG expression. So this seems to suggest that spite does have a negative effect on IgG growth, whereas glutamax has a positive effect, and the combination then lowers the effect of the glutamax. Another example would be the addition of spite to the two different buffers. So when we add spite to hippies, and that's the dot, this in the middle, the spot in the middle shows that there's absolutely no effect, between spite and hippies. But when we add spite to a media that contains the bioreactor buffer, we now see an increase in the IgG growth. So the interaction of spite with the bioreactor media essentially improves or increases or acts as a positive factor to IgG expression. A third example would be where you have more than two interactants. So in this case, we are adding spite to a system that already has glutamax, and as you can see, the first two dots would be the effect of spite with glutamax as was shown in the first diagram, where there's a negative effect. But when we add a third component such as hippies buffer, again, we don't see much of an improvement. But if that buffer in this case was bioreactor buffer, we do see now that suddenly the IgG growth then increases. So a combination of spite, glutamax with bioreactor buffer now has a positive effect on the growth, of the growth media condition. So these three graphs are a very big testament to the power of a full factorial DOE in being able to identify what the best or the optimal conditions would be for any cell line development. As a summary of what we found with the IgG quantification in the octet, what we see is that the different additives have different effects, with polyamine and glutamax having a positive effect on IgG growth, whereas bioreactor buffer itself, would have a negative effect on IgG growth. Spite also reduces the positive effect that we see with glutamax, but then the combination of spite, the bioreactor buffer, and glutamax eventually then does have a positive, effect on the IgG growth. So this kind of analysis was done for all the interactions, and what we saw is that by far a lot of the other factors or combinations had negligible effects, if any at all, on the IgG production. So in summary, what we saw is that if we only looked at the IgG production from the basal optical media alone, we do obtain a figure of about 8.8 micrograms per ml. We also saw that the highest condition, the best interactants, produced as high as 14.6 micrograms per ml in IgG production, whereas the lowest condition gave us, a reduction of about 10% from the basal optical production of 8.8 to about 7.8 micrograms per ml. We also could do a prediction based on the statistical modeling. And what we predicted was that a combination of polyamine, spired glutamax, and bioreactor buffer would produce as high as 13.6 micrograms per ml. In reality, we did get the highest condition being 14.6 micrograms per ml. So essentially, we also did predict that if you just looked at the basal optical media alone, the model would give you a prediction of 9.6 micrograms per ml, where in reality, we did get only 8.8 micrograms per ml. So the prediction was about 42% higher than what we actually, obtained using optical media. So this design of experiment, is a fairly good way to run a fairly quick experiment using two integrated systems, the BioMech workstation and the Octet HTX, to be able to look at 96 different, interactions and be able to determine which particular interactions or supplements would not necessarily give you any value in your cell line development, and therefore would be a waste of time and a waste of money during the development process. So to look at other things now that we did beyond the IgG growth, I'm going to turn it back over to Mike to continue with the presentation.
Thanks, David. Yeah, that was a great explanation of how we were able to achieve measurements on the IgG production, which was still the main goal for the optimization process. But we did want to take it one step further and see if we could establish any sort of mechanism by which these factors were having their effect. And you can imagine easily two different ways that you could get an increase in IgG production. One would be essentially to stimulate cell growth, so you have more cells expressing the same level of protein, or it could be that you're inducing more protein production, so essentially the same number of cells, but each of them producing more IgG. Ideally, if you can discriminate between these two effects, you might have the potential to harness both of those, such as by increasing the number of cells initially and then switching over to more of a protein production model by changing the additives. So for this, we turn to the SpectraMax i3X with Minimax imaging cytometer, shown to the right, and this is from Molecular Devices. The blue portion of the instrument is the SpectraMax i3X, which is essentially a plate reader that allows you to measure standard measurements such as absorbance, fluorescence, or luminescence at the well level. And if you add the orange component, the Minimax imaging cytometer, this allows you to take 4x images in either bright field or two different fluorescent wavelengths. So you can really get per cell data rather than just well level data. We actually took two approaches to measuring cell growth. One was to just use standard bright field cell imaging, and another was to use an absorbance assay by the addition of XTT. This is a reagent that will just eventually turn into an orange color, that's based on the number of cells per well. It's a very straightforward assay. It's just add, incubate, and read, and we automated this process on the BioMech as well. And we'll discuss why we use both of these approaches. So shown here is the first image of our CHO cell lines that are grown just in standard OptiCHO media. And what you can see is that the cells essentially form 3D colonies in this condition. This could potentially result in undercounting cells if only using an imaging assay because of the nature of the colonies. However, we were able to see some differences with the different additives in that the presence of glutamax resulted in increased sizes of these cell colonies, suggesting it does have an effect on cell growth. We also noticed that the addition of spite seemed to increase the number of individual cells between colonies. So certainly just looking at these images, you can imagine we saw higher cell counts than we did in the OptiCHO media alone. And if we combine those two, spite with glutamax, you essentially see both effects, larger colonies as well as more cells in between the colonies, resulting in higher cell counts. To just kind of summarize what that looks like across the board, again, this would suggest that glutamax and spite, those two factors which we've already seen can have positive effects on IgG, might likely be working through a cell growth mechanism. This also implies that things such as polyamine, which did not seem to have an effect on cell growth, might be working through a different pathway, such as increasing protein production. And again, this might be a way of perhaps staggering these additives to increase your production even further. We also, as I mentioned, used the XDT assay as another surrogate for cell growth, simply because we figured this should not be affected by the 3D morphology. However, because we have different additives in different wells, there is the chance that the chemistry itself could be affected somehow by the additives. The good news is we saw a high degree of similarities in that glutamax had an increased effect on cell counts in either method, cell imaging or XDT assay, suggesting that this really does reinforce the idea that glutamax is inducing cell growth. The same is true for the spite plus bioreactor buffer that we saw originally had the positive effect on IgG titer as well. However, we did see some differences compared to our cell imaging, for the one example being efficient feed A, which basically had no effect on IgG titer and perhaps even a negative effect on cell counts by imaging, actually had a positive effect on XTT, suggesting that we did have some disparate results, and we might need a third assay to try and distinguish between these two. One way to do that might be to trypsinize the cell colonies and either replate them for additional imaging or possibly count them through another mechanism such as flow cytometry or other cell counters. So that pretty much sums up the data that we had for this level of IgG optimization through changing the media components, but there were some caveats that we learned through this process. One is that, as one might expect, evaporation can significantly impact your results. So the data we showed for IgG titer was from a 4-day incubation, and because we're using the 384 well format, the edge effect, where evaporation is most prominent on the outer wells, can have a significant impact. That's summarized by looking at the CVs of our replicate wells. If we only look at wells that had all 4 of their replicates on the interior of the plate, the average CV across samples was only 2.9%, a remarkably low level for this type of biological assay. However, if we compare that to the octet readings of our quadruplicate values, where some of the wells were on the edges, the outer portion of the plate, you can see that CV increases to 9.6%, indicating there was a significant edge effect on this plate. And for this, the edge wells were excluded from the calculations, and that's one more reason why it was very relieving to have the statistical software be able to predict the models so that you're not entirely dependent on a single data point for your analysis. We also found that some of the additives can affect the octet baseline. One example is linoleic acid, oleic acid, and albumin. There was a combination of these 3 factors that we thought about adding, but we noticed that there was a spike in the baseline every time these factors were added, and because they would not be present in every well, it would be very difficult to discriminate real from false positive effects on the IgG production. So because of this, this was eliminated from the screen. And we've already touched on some of the caveats we saw with the cell imaging and XDT in that morphology can possibly play a role in getting misread cell counts, as well as the different additives could be affecting the XDT chemistry, or evaporation can also have an effect. So that's another reason why we wanted to have these somewhat complementary yet also somewhat redundant ways of measuring cell growth. So to bring it back to the process of cell line development, again, we were really only demonstrating one aspect of a much longer process, really focusing on the optimization step of the workflow. And we showed how each of the 3 components, the automation for sample processing, the imaging or absorbance reads, as well as the protein production and protein quantification reads on the octet, really enabled this step to be put through at a relatively high throughput. Again, for such fast processing and fast analysis, we could have increased this process even further to include additional factors or additional concentrations with relative ease. However, what's important to note is that all of these instruments in some way, shape, or form can be used throughout the entire cell line development workflow. Such examples include using the octet to do the protein production assessment of large numbers of monoclonal wells, as well as using an imaging, whether it be the I3X with Minimax or other imaging components, to track cell growth and to establish monoclonality during the process as well. The automation can certainly be used to automate each and every one of these components to basically enable whether it be a cell line development or any of the sample processing needed for the type of analyses we've discussed. So the throughput concern has certainly been answered with each of these instruments. But if we want to look at the sample and data integrity, what really becomes more powerful is that you can actually integrate these devices all together to ensure that samples are processed from beginning to end of this workflow in one continuous fashion. And one example of a fully integrated cell line development system is shown to the lower right, where the biomech is once again there to prepare samples, plate cells, transfer media for analysis, and then the analyzers can be integrated with a robotic arm, and those plates can be then processed on individual analyzers and sent back to the incubator for additional incubation. Not only does this ensure sample integrity because there's less need for manual intervention, but each plate can be barcoded to ensure that every plate is being processed at a known time in a known fashion. We also have a software component to the idea of data integrity. So as the samples move through the process, our DART data tracking software can basically append each new piece of data as it's generated to each plate to ensure that you don't have to go back and try and figure out what plate corresponds to what sample, which corresponds to which data. All of that is brought forward throughout the process. And in fact, that data, as it is learned, can be used to drive additional pipetting. One example would be, as you identify which clones have your highest producing IgG, the biomech can take this information and then automate the replating and expansion of these cells into larger well plates. We also have a software solution for the challenge of scheduling campaigns. As I mentioned, you can go from very low throughput, where you're maybe transfecting 1 to 2 wells, all the way up to very high throughput, where you're screening hundreds of thousands of clones to establish monoclonality and IgG production. But because you have to have a system that can handle the maximum sample number, you also want to find ways to stagger these campaigns so your system is getting the maximal usage. And our SAMI scheduling software is the method by which you can establish what samples are currently on the system, when the automation or analytical devices are going to be in use, when samples will be coming off of the system, and when you'll have more room in your incubator, for example, or whatever your rate limiting step may be. So again, just another way that you can get the full maximal power out of your fully integrated cell line development system. So with that, we'd like to wrap up. Hopefully we've demonstrated how we used our design of experiment approach to identify positive reagents for IgG production, as well as some pretty unique interactions that would not have been detected had we not been using this design of experiment approach. And we were able to identify conditions that should increase our IgG production between 42 and 66%, whether you're using the statistical modeling software or whether you're using the actual output numbers. Obviously, partnerships were key for this type of work. To have expertise in automation, protein production measurements, and imaging or other types of analyses really bring together this project. And finally, we showed how integrating these devices, the Biomech, the Octet HDX, and the SpectraMax i3X with Minimax or other imaging instruments can really enable a complete cell line development workflow, and how samples and data can then be tracked throughout the system. For more information, you can visit biologicsbydanaher.com. And then we've got a few disclaimers that we need to put up there. And after that, thank you very much for your attention, and we're happy to take questions.
Thank you, Dr. Kowalski and Dr. Apiyo, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, you will receive a follow-up email after the presentation. For the Q&A portion, we will also have Sean Alvarado, Field Application Scientist from Molecular Devices. Let's get started. Our first question is, for Sean, what automatic measurements can you make with the Minimax imaging cytometer without adding fluorescent dyes?
So one of the best features about the Minimax imaging cytometer is the ability to do the stain-free analysis. So if you're looking to do cell counts, if you're looking to assess confluency, using our field analysis tool, you can do classification with morphology. All this can be done without any dyes. So when you use the Minimax, you can actually take your cell line, perform the initial analysis telling the system what is a cell, what is not a cell, and then further you can classify based on morphology. For example, if you're doing an infectivity assay, you could select cells that are infected and uninfected. And once you set this up for the cells that you're going to be doing, you can actually save this as a protocol to be used every time you're going to run the assay, making the process automated and not requiring any dyes.
Thank you. The next question is for David. What's the specificity, sensitivity on these instruments?
Yes, thank you for the question. So the specificity for the octet instruments or the octet biosensors would depend on what your analyte, your biological molecule of analysis, is. So, for example, if your target analysis is an IgG, such as we have talked about in this webinar, then your biosensor would potentially have a protein A or protein G, on it. The biosensors that we currently sell use recombinant protein A or recombinant protein G, where the potential BAC or albumin binding epitopes have been removed. So they should be specific to any FC region of an IgG or potentially, fusion proteins. So the systems are generally specific, but just like with any given biological assay, sometimes one may need to assay develop to remove potential non-specific, binding. When it comes to sensitivity, all the octet instruments that I talked about today should be able to measure as low as 0.05 micrograms per ml in a direct assay. So a direct assay, again, is one where you're dipping the sensors into the reagent in a one-step assay. However, if you have concentrations lower than 0.05 micrograms per ml, then you can build the assay in what we call an indirect assay, such as a sandwich ELISA assay, where you amplify signals using polymerization chemistries. In that case, you can lower the sensitivity to as low as 10 picograms per ml when doing a quantitation assay.
Thank you. The next question is for David. Does the octet quantitation approach apply only to IgG samples? If not, what might the process look like for a recombinant protein?
Yes, great question, and thanks again. So IgG quantitation is what we did in this webinar. However, the systems can be used for any type of quantitation so long as you have a capture molecule. And by that, really what I mean is that if you had a recombinant protein, for example, you could potentially immobilize a specific antibody towards that particular protein and use that newly customized biosensor to be able to do direct quantitation. So it's not quite the case that it's only for IgG quantitation. It can also be used for recombinant proteins, but in that case, you would have to develop what we call customized biosensors. Those customized biosensors, once developed and stored, could then be used in direct assays where they're being dipped, simply into the analyte for a one-step assay.
Thank you. The next question is for Mike. Can automation be used to optimize culture growth conditions of bacterial expression systems, for example, E. coli, for the parameters such as temperature, time of harvest, shaking speed, and OD?
Yeah, that's a great question. It's a similar application in the sense of you might be looking for something similar for protein production, but very different in the sense of now dealing with prokaryotic cells. From a liquid handling perspective, they're very similar, right? You're still moving things from one position to another, but one consideration might be a lot of times people working in bacteria are more used to, say, round-based plates such as Petri dishes. Ideally, you can do things in an SBS format, so basically standard micro titer plate formats. perhaps using deep well plates to get some of your liquid culture. Or, as I mentioned before, the span 8 pipetting system does work well with tubes. So if you're doing things in a shaking tube type of platform, that would also work. As far as changing temperature, that's one thing to consider when it comes to what you're integrating or what some devices you might include. Obviously, if you're working with multiple samples in one plate, that plate can only be incubated at one temperature. But you could have smaller incubators or even some on-deck heating and cooling, since you wouldn't necessarily need the CO2 typically associated with mammalian cell culture. And so we could do some of that incubation and shaking on deck or through smaller integrated incubators as well. So long answer to a short question is yes, you definitely can optimize that type of workflow with bacteria and using a lot of the same devices, heating and cooling. You could measure your OD with a plate reader or other such analyzers. So definitely would be feasible.
Great, thank you so much. The next question is for Mike. Were these experiments performed with adherent or suspension cells?
Yeah, that's also a great question. So CHO cells are obviously able to be grown typically under either condition, suspension or adherent. And I would kind of call these loosely adherent. What we found is that after multiple passages in the OptiCHO, the cells, even if we started them in adherent culture, would eventually switch to suspension. However, we were doing this in the first 1 or 2 passages in this media, so they were still adherent to the bottom of the plate. We have been doing some similar work for cell line development working with suspension CHO cells, and depending on the plate you use, the workflow is very similar. But if you want to take an imaging component, it does help to work with some plates like the CoStar, I forget what they're called, but it's basically a plate that loosely binds some suspension cells, but they can be easily released from the plate just through pipetting. So again, the system should work with either. It's just you need to take a couple things into consideration depending on the analysis that you're using. But the octet would be unaffected or should be unaffected by the presence of cells. So if some do get transferred, it should not affect your reads. And again, the imaging, there's ways that you can have the cell settle to the bottom of the plate and stay loosely adherent for more accurate imaging.
Thank you. The next question is for Mike. Wanted to know if it was possible to build or Beckman has a custom integration for the octet or are potentially working on one for this system.
Yes, we do. So in the image that I showed, it's obviously very difficult to see in a small image, but the cell line development system that we demonstrated did have an integrated octet, and we've demonstrated that in the past. It is able to be directly controlled through the automation where, like I said, a robotic handler would place the plate in the octet for the read and basically essentially push the go button. The data would then be sent back to the automation, and as I mentioned, that data could be directly used to drive hit picking or other applications on the instrument. So yes, the Octet HDX and as David mentioned, I believe the 384 have both been integrated with Biomech liquid handlers.
Thank you. The next question is for either David or Mike. Was there any analysis of how unattached culture would affect the cells?
Yeah, so I can start with that one. I think we sort of touched on that earlier. As I mentioned, what we did was using adherent cultures, but again, the cells can be grown in both suspension or in adherent cultures. As I mentioned, some of the work we're doing now is with suspension cultures. I've not done a direct comparison. We've actually changed to a different media. We're using CHO growth A from molecular devices for the suspension cultures, and the cells are still expressing IgG, but I've not done a direct comparison to how we did this type of analysis. But the same rules would apply. You would certainly be able to do a very similar approach for optimization and then just plate these suspension cells on top and still process them using the octet. David, anything to add?
No, I think you're correct. I don't think I have anything to add to that.
Great, thank you so much. The next question is for either of you. Is the effect stronger if you see glutamax plus the spite and buffer than just glutamax? I noticed there was a difference from the baseline, but was not clear on this difference.
I think so. I think there was a fairly slight difference. And Mike, you can chime in here. But what I did notice is that indeed, when you had glutamax plus spite and the bioreactor buffer, you see a fairly slight improvement from when you just have glutamax alone. The question in my mind is whether the difference is significant enough to justify the cost of adding spite and the bioreactor buffer as opposed to just having glutamax by itself. So that's an analysis that I'm not sure we've done, Mike, but there indeed was a very slight difference between the three combined versus just glutamax alone.
Yeah, David's exactly right. So I've pulled up slide 13, which hopefully can roughly demonstrate that the green line in the third graph is higher than the red line in the first graph, so there was an improvement with the spite plus bioreactor buffer according to our statistical analysis as well as our actual readings. But David is correct that you would have to do the kind of cost-benefit analysis of is this worth the additional cost of the reagents, and we didn't necessarily look at that. But yes, there was an improvement with all of the factors that, as we mentioned, the predicted best condition would be glutamax plus polyamine plus spite plus bioreactor buffer. But whether you can get most of the way there with just glutamax certainly seems to be the case. So again, to do the kind of cost-benefit analysis of what the reagents cost would definitely be worthwhile looking at.
Thank you. We'll wrap up with one last question, and that question is, for Mike, which software tool was used to design your DOE and generate biomech scripts?
So two different questions there, I guess. The first one is that we use Design Expert from StatEase, but as David said, you can use a variety of different statistical software for this type of application. In the past, we've had a software that was used to then directly translate that into biomech script. That was not what I did here. This was basically just I developed a list of factors and then essentially figured out the best way to put them on the plate so that they would necessarily work in a reasonable fashion to allow me to get to a 96-well format. So there could be some tools that can help you with the statistical design software. And then again, to create the biomech script or the biomech method was basically just things that I worked out the layout in my head and just wrote in a few different transfer steps.
I would like to once again thank Dr. Kowalski and Dr. Apiyo for their presentation. Do you have any final comments?
For myself, just thank you again for attending, and certainly if there's additional questions we haven't gotten to, we'll certainly try and follow up with those. But yes, thanks very much for the opportunity, and thanks everyone for attending.
Yes, I would also like to thank everyone for attending and the opportunity to be able to speak to you all about this webinar. I'd like to add that if you do need any more information on how the octet works or BLI in general, please let us know. Thank you.
Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.